Hello, everyone, and welcome to today's webinar, Advancing Towards Personalized Medicine in Lung Cancer, a focus on gene rearrangement testing. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott Scientific Affairs Molecular Diagnostics. To learn more, visit molecular.abbott.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow that process to obtain your credits. I now present today's speaker, Dr. Yelina Noskina, the Global Scientific Affairs Manager at Abbott Molecular. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Yelina, you may now begin your presentation.
Thank you, Sabrina, and thank you everyone for joining me and listening to my presentation on advancing towards personalized medicine in lung cancer, a focus on gene rearrangement testing. These are my disclosures, and I would like to start by talking about the fact that identifying specific biomarkers associated with specific types of cancer has become a driving force in the advancement of personalized medicine, and there have been various technologies developed in order to do so. Gene rearrangements are a prime example of such biomarkers, having been identified as a common genetic alteration that can be targeted by various therapeutics, especially in non-small cell lung cancer. Therefore, identifying the presence of gene rearrangements is the objective of various companion diagnostic assays. This review will focus on the emerging technologies to identify gene rearrangements, specifically focusing on ALK and ROS1 in non-small cell lung cancer. I will provide a technical overview of the methodologies associated with identifying gene rearrangements, including a review of the various guidelines and recommendations, the common practices and the difficulties associated with testing, and finally discuss the best course of action that can be taken through the patient's treatment cycle. But first, I would like a quick review of the global statistics surrounding lung cancer. In 2018, lung cancer was designated as having the highest number of incident cases and deaths globally. Non-small cell lung cancer makes up 80 to 85% of all lung cancer cases, with the majority being adenocarcinoma type. The 5-year overall survival rate of non-small cell lung cancer is approximately 24%, while the 5-year metastatic lung cancer survival is as low as 6%. In general, there have been limited therapy options when it comes to non-small cell lung cancer. Prior to 2009, chemotherapy was still sought as the best course of treatment for patients with advanced non-small cell lung cancer. And in 2006, epidermal growth factor receptor inhibitors like gefitinib were considered novel agents for second-line therapy without a clear definition of which EGFR abnormality was critical. But in 2007, the fusion between the anaplastic lymphoma kinase and EML4 genes was identified in lung cancer, and the result of the fusion between the two genes was a protein with increased tyrosine kinase activity. This fusion protein and its increased activity could be considered a potential target for non-small cell lung cancer therapy. In 2009, the American Society of Clinical Oncology released guidelines for stage 4 non-small cell lung cancer with the following recommendations: that gefitinib could be used as first-line therapy for stage 4 non-small cell lung cancer patients that had an activating EGFR mutation, yet the guidelines still stated that there was insufficient evidence to recommend routine use of molecular markers. Instead, use of molecular markers was supported to obtain a more accurate histologic classification of the disease or for investigational purposes. But in 2010, the use of a new tyrosine kinase inhibitor, crizotinib, was shown to be associated with increased survival of non-small cell lung cancer patients with tumors positive for ALK gene rearrangement. And in 2011, the FDA concurrently approved Pfizer's Xalkori and Abbott's Visus ALK break-apart FISH probe kit, being the first example of a co-development of a companion diagnostic with the associated therapeutic. This led to the College of American Pathologists, the International Association for the Study of Lung Cancer, and the Association for Molecular Pathology to release the first molecular testing guidelines for non-small cell lung cancer in 2013. Here they defined the best practices in molecular testing for therapy selection in advanced non-small cell lung cancer patients, specifically recommending that testing patients for EGFR mutations using PCR-based technology to select them for gefitinib and other EGFR tyrosine kinase inhibitors, and testing for ALK gene rearrangements using a dual break-apart FISH probe to select patients for crizotinib tyrosine kinase inhibitor therapy, with the possibility that an ALK IHC test, if carefully validated, could be considered as a screening methodology for selecting specimens for follow-up ALK FISH confirmatory testing. In 2014, ASCO issued follow-up guidelines which reaffirmed the CAP IASLC and AMP guidelines and added the recommendation that if an institution is not using an FDA-approved assay, then they should develop a specific testing algorithm, and that IHC could be used as a screening method followed by the FDA-approved Visus ALK test for confirmation. By 2018, ROS1 was added as the third molecular target to be considered mandatory for testing for targeted therapy selection, with more potential targets being discussed and studied for potential therapy selection. All three of these biomarkers have an effect on downstream signaling, specifically on cell growth and proliferation, and at the diagnostic level, they can provide predictive and prognostic information, which is why we should take a closer look at these. Tyrosine kinase receptors, or receptor tyrosine kinases, which are cell membrane proteins that act as signal transduction regulators, make up our EGFR, ALK, and ROS1s belong to this group. These are activated when the cell surface receptor binds to specific signaling molecules, known as ligands, which activate the tyrosine kinase domain, thus sending signals that affect cell proliferation, differentiation, migration, and apoptosis. Abnormalities in these proteins can result in deregulated tyrosine kinase activity, leading to unregulated cellular activity. But at the same time, these proteins can be targeted by tyrosine kinase inhibitors. And when there is a specific inhibitor targeting a specific protein, the outcome is personalized therapeutics. Being able to identify a genetic abnormality affecting the tyrosine kinase receptor is key to determining the appropriate action to take in blocking the abnormal activity. For instance, EGFR is known to have specific mutations, while gene amplifications are commonly associated with a MET receptor tyrosine kinase. But the genetic abnormality we will focus on today is gene rearrangements, as this has been identified to be the most common abnormality associated with both ALK and ROS1 genes. In general, chromosome rearrangements are not uncommon and have been categorized as demonstrated here as inversions, duplications, deletions, and translocations. But the overarching point is that when specific genes are involved, they can result in abnormal fusions between the regulatory elements of one gene with a coding region of another gene. If the involved genes are receptor tyrosine kinases, then it is possible that the tyrosine kinase portion of the gene can be fused with a regulatory region of its fusion partner, resulting in a chimeric protein with constitutively activated tyrosine kinase, basically a tyrosine kinase protein that does not require activation by a signaling molecule. In non-small cell lung cancer, the first gene rearrangement was identified as being between ALK and ELM4 genes, but soon after, other partners were identified. These rearrangements result in constitutive activation of the ALK tyrosine kinase activity, which could then be targeted by a tyrosine kinase inhibitor, such as crizotinib. An interesting aspect to keep in mind when it comes to ALK gene rearrangements is that the breakpoint region of ALK appears to be consistently at or near the transmembrane portion of the gene, leaving room for variability in the resulting fusion gene. That is, it can have variation in the cellular localization and level of expression. And when it comes to that variability, according to the Atlas of Genetics and Cytogenetics in Oncology and Hematology, there have been over 50 fusion partners identified for the ALK gene. Of course, not all of these occur in non-small cell lung cancer, but variations of those partnerships and more partners continue to be revealed. Besides ALK, there is another gene that has been shown to be associated with non-small cell lung cancer, and it is also a receptor tyrosine kinase. The human CROS oncogene 1, or ROS1, has structural similarities to other receptor tyrosine kinase proteins, like ALK. With ALK, it is also has sequence homology, which allows it to be targeted by the same tyrosine kinase inhibitors, such as crizotinib. But unlike ALK, ROS1 is normally expressed in many tissues, such as kidneys, the cerebellum, peripheral neural tissues, the stomach, the small intestine, and the colon. The rearranged gene was first identified in 1987 in glioblastoma, and a fusion partner was identified in 2003. But it wasn't until 2007 when Rykova and associates identified ROS1 gene rearrangement in non-small cell lung cancer. Like ALK, ROS1 has been associated with over 25 partner genes and in many tissue types, in some cases only being identified in one patient. So how can biomarkers be identified so that appropriate decisions can be made at the care of the patient? In general, there are various methods to identify biomarkers. One can detect the presence of the gene or genetic abnormality using cytogenetics and cytohybridization, such as FISH, or KISH, sequencing such as Sanger sequencing and next-generation sequencing, or polymerase chain reaction amplification, PCR or RT-PCR. Another approach is to identify the actual product of the abnormality. This typically involves using antibodies targeting the product via IHC or ELISA methodology. There are also other methodologies such as flow cytometries and various microarrays. But when it comes to identifying gene rearrangements, the best approach has not yet been decided. Initially, there were three main methods used to identify ALK gene rearrangements in non-small cell lung cancer. There was FISH, IHC, and PCR amplification. FISH is looking at the gene pattern. It is looking for the presence or the absence of a gene or the relative location of the gene in individual cells. In the case of ALK, this is looking at the presence or it's not looking at the presence or absence of the signal, but looking at the relative location of two signals to determine whether a gene is still intact or if it has broken apart. IHC is looking at protein expression. It identifies the presence of a protein or a portion of the protein that is targeted by the antibody in the tissue. In the case of ALK rearrangement, the target would be the tyrosine kinase portion of the protein. In PCR amplification, we can identify genetic variation to the single nucleotide level, making it a great technology for identifying gene mutations or gene rearrangements if the exact location where the rearrangement occurs is already known. And while the ALK-FISH assay was chosen as the companion diagnostic for Pfizer's Xalkori, due to the fact that IHC and RT-PCR missed some of the positive cases during the clinical trials, efforts continue into the improvement of IHC assays in order to match FISH capability. Many studies were conducted using several antibody clones for the ALK protein, with the outcome that Novocastra, the 5A4 clone, and the Ventana D5F3 clone were selected as optimal choices to match FISH capabilities. In 2016, an analysis by Jang and Associates was performed on 12 different studies, which evaluated testing methods with almost 4,000 non-small cell lung cancer specimens in locations throughout the world. They concluded that using IHC to screen non-small cell lung cancer cases was a cost-effective method when a 4-tier scoring system such as that recommended by Novocastra 5A4 ALK test was used, where the 1+ and 2+ results would then be reflexed to ALK-FISH analysis for confirmatory testing. The outcome of their data, along with further analysis, which adds several other studies to this assessment, allows us an overall assessment of the sensitivity and specificity of the two antibody clones as compared to FISH, finding that in most cases, IHC was either not as sensitive or as specific as FISH methodology. Taking a further look at the FDA-approved IHC test demonstrates that even in their studies conducted specifically for FDA approval of the Ventana D5F3 IHC assay did not reach the 95% agreement to the FDA-approved FISH test. This range in concordance observed with IHC could be due to several factors. As mentioned by Aruda and Minas-Knudsen, when it comes to IHC to identify ALK rearrangements, there are risks of false positive and false negative results caused by the relatively low expression levels of rearranged ALK in non-small cell lung cancer, thus requiring amplification systems to enhance ALK detection, and the morphological features of the tumor that may interfere with those amplification systems. There's also the inherent lack of assay quality controls when it comes to IHC. And with the two leading clone manufacturers, there is no standardized method for assay assessment, where the interpretation associated with using the D5F3 clone is significantly different from that of the 5A4 clone. While FISH methodology has been associated with difficulties due to pre-analytical method variability and the need for experienced technologists, these same factors can be attributed to IHC methodology. Therefore, consideration should be taken interpreting IHC results with caution, and employing confirmation by FISH could be beneficial when facing challenging cases. Overall, when comparing FISH to IHC in identifying ALK gene rearrangements, the following factors can be considered. Both are slide-based systems requiring similar sample processing methodologies, such as sample fixation and paraffin embedding, and even placing the tissue samples on the slide. When it comes to FDA approval or other regulatory body approval, the FISH assay is FDA approved and CE marked, and the Ventana ALK D5F3 CDX assay is also FDA approved, while the Novocastra clone 5A4 CE marked assay is only CE marked, not FDA approved. When it comes to the special equipment required for those assays, there is a requirement of having a fluorescent microscope to perform FISH analysis, as well as an automated slide warmer can be beneficial. The Ventana Bond Max is required equipment to perform IHC using the D5F3 clone, while Novocastra does not have any such requirements. At the same time, the automated slide processor is an option when performing FISH methodology. When it comes to training requirements to perform these assays, FISH does require the training on on recognizing special signal patterns and the cutoff of the assay, while Ventana has an interpretation guide to assist pathologists in understanding the different types of staining patterns that can be observed and whether or not they would be positive or negative, while Novacastro relies on the standard intensity level assessments of 0 to 3+. And when it comes to establishing result reporting requirements such as scoring and cutoff levels, the FISH assay comes with a specific 15% positive out of 50 tumor cells as a scoring requirement for a positive case. While this is varied for IHC based on processing and signal detection amplification systems with the two clones, where Ventana relies on a positive-negative system based on the interpretation guide, and Novacastro relies on the 0 through 4+ through 3+ intensity staining levels, even recommending to confirm with another method when the results are 1+ or 2+. Interestingly enough, differences between these methodologies for identifying ROS1 gene rearrangements have been more clear-cut. FISH has historically been considered the gold standard in identifying ROS1 gene rearrangements. Again, this technology does not require knowledge of the fusion partner when using break-apart probes, and it can be performed on formalin-fixed, paraffin-embedded, and cytology specimens. But when it comes to IHC, since ROS1 is normally expressed in multiple tissues, using IHC becomes more challenging than other assays associated with fusion proteins, as this methodology may not be able to distinguish between normal expression and increased expression due to amplification versus gene rearrangement. An assessment by Pal and Can of multiple studies comparing IHC to FISH for ROS1 showed variability in the results. They found that detection systems, the antigen retrieval methodologies, and staining conditions affect outcome, along with a variability in scoring systems. And in most studies, demonstrated a high sensitivity but a variable specificity. Therefore, IHC has been presented as applicable for pre-screening purposes with a follow-up with confirmatory FISH testing. Looking back at the guidelines, we can see that there has been continued development in the treatments of non-small cell lung cancer. Between the 2018 CAP, IASLC, and AMP updated guidelines, which added ROS1 gene rearrangements to be considered part of the absolute minimum primary biomarkers to be tested on patients with appropriate non-small cell lung cancer profiles. This was due to the fact that ROS1 was shown to also be a target for ALK tyrosine kinase inhibitors. The specific guidelines on the appropriate methodology stated that EGFR mutation should be tested by PCR methods. ROS1 gene rearrangements are to be performed by FISH testing, and ALK gene rearrangements are to be tested using FISH rearrangement probes, and they added a properly validated IHC method as an acceptable alternative to FISH testing. This was if the evidence supporting the use of IHC was adequate and when the IHC results are clearly positive. But challenging to interpret cases should still be followed up with another test such as FISH. At the same time, the guidelines included using multiplex genetic sequencing panels for next-generation sequencing. They introduced this methodology as the preferred option to identify other treatments beyond those of EGFR, ALK, and ROS1. The following year, the National Comprehensive Cancer Network added BRAF mutation analysis to the list of primary molecular biomarker analysis with methodology guidelines similar to those presented by CAP, IASLC, and AMP. They also included the FDA-approved IHC Ventana D5F3 CDX assay, and they recommended broader molecular profiling using NGS, but did not advocate any specific assay stating that not all alterations can be detected in a single NGS assay. This year, NCCN added MET exon skipping and RET gene rearrangements to the growing list of key molecular biomarkers that should be tested, still encouraging the use of broader molecular profiling, but again, not advocating for any specific test while cautioning on the choice of assay since not all alterations can be detected by the individual test, implying that there may be a need to perform more than one NGS assay to answer all questions. So what should we know about NGS testing? This is a high-throughput method that performs simultaneous sequencing of multiple targets in multiple samples. It starts out with a library of DNA fragments is created. These fragments are then amplified and sequenced. The sequencing software analyzes the fragment sequences, creating larger constructs, and the constructs are further analyzed by bioinformatics software, comparing them to known or normal sequences. This methodology is associated with a wide breadth of applications. Whole genome sequencing can be performed, whole exome sequencing, cDNA sequencing and gene expression profiling can be performed using NGS, as well as methylation sequencing and mapping, and many more applications are available. This NGS provides the ability to identify genetic alterations on a much larger scale, thus driving advances in clinical research. NGS started at the academic and research setting, and it has been entering the clinical field over the past several years, developing in private institutions, and it has even been growing in the community-based practices. In one study, use of NGS in the clinical setting was seen to increase at a community-based practice by approximately 15% in 5 years, and it continues to grow. As mentioned earlier, numerous NGS assays for research use only have been validated in laboratories with variations in the sensitivity and specificity, the amount of DNA required, the percent of tumor tissue needed, the limit of detection, and the success rate. But in the clinical setting, NGS can provide information on targetable mutations and direct patients with limited therapeutic options into clinical trials. With the high complexity of this technology, though, chances for variability are also high, which has led to a measured implementation. As a quick overview of the general complexity of the methodology, it can be seen here with the acknowledgement that it is a continuously developing technology. It requires multiple steps and multiple days, starting with nucleotide isolation, followed by target enrichment, where different methodologies can specifically identify the areas of research or testing, such as hybrid capture or amplicon PCR, when specific targets are amplified for further sequencing. Once this is completed, library construction is performed where these targets are appropriately labeled, and then massive parallel sequencing ensues, followed by comprehensive data analysis relying on bioinformatics algorithms, which can be experimental, and dependence on the reference sequences and whether they are public or proprietary databases. Many quality checks are required throughout this workflow. There are, of course, advantages to using NGS in the clinical setting. Benefits such as expanding options and providing more information on initial tumor profiling and therapy management. Adoption in the clinical laboratory setting has been increasing, especially with the limitation of tissue available and the increasing list of molecular targets providing therapeutic guidance. Increasing demand for as well as increasing demand from the oncologist and the patient. The clinical utility is increasing as target enrichment and improved methodologies decrease time to result. What initially started as several weeks is decreasing to several days. But there are, of course, drawbacks. Extensive information is included with results of undetermined clinical significance, thus being criticized for providing clinician results beyond those that are actionable. There's also limited capabilities in identifying gene rearrangements. Using targeted enrichment requires prior knowledge of the potential fusion partners, while higher assay complexity and lower success rate has been also a factor based on the availability and the quality of sequencing data, often due to processing issues. The longer time to result and higher costs can also be problematic when patients are waiting on treatment decisions and there may be a risk of not getting insurance coverage. And when it comes to gene rearrangements, specifically with ALK having more than 50 potential fusion partners and ROS1 having more than 25, standard NGS is unlikely to identify novel or rare rearrangements if not performing RNA-NGS. As mentioned earlier, there is a growing number of laboratories bringing NGS in-house with variation in methodology and assay parameters. Therefore, when it comes to using NGS specifically to identify ALK gene rearrangements, I will focus on the 3 assays that have received FDA approval or authorization: the MSK Impact, Foundation 1, CDX, and the Oncomine DX assay. The MSK Impact assay looks at over 300 genes, but only the EML4 ALK translocation. It is also an institution-specific assay that requires sending of the sample to Memorial Sloan Kettering. The FDA-approved Oncomine DX assay is produced by Thermo Fisher Scientific and is available for laboratories to purchase and implement, but it has no claim in the U.S. for ALK gene rearrangement. While the FDA-approved Foundation 1 CDX assay, also an institution-specific assay, showed only 92% accuracy for positive ALK cases compared to FDA-approved FISH and IHC tests combined. Compared to FISH and IHC sensitivity, NGS assays have been seen to be as low as 80% with the expected limitations of less common rearrangements. Similarly to ALK, depending on NGS method used, the exact known fusion partners of ROS1 may be identified. Using RNA as a starting material may provide more information depending on the assay design. There are multiple examples where a unique fusion partner was identified in only one patient sample, usually through whole genome sequencing. And this is a very good methodology for research purposes and clinical trial stratification. But the sample quality and quantity is even more critical for successful RNA-NGS results. As mentioned, the higher failure rate that has been associated with NGS could be a significant factor when it comes to choosing the appropriate methodology. And as can be seen here in a study that evaluated FISH compared to RNA-NGS and DNA-NGS, it is significant to note that RNA-NGS showed a 47% failure rate based on the quality parameters and overall ability to obtain significant amounts of RNA. Diagnosing patients with non-small cell lung cancer requires a histological assessment of the tumor and the continued efforts to have minimally invasive procedures and reduce patient discomfort lead to the diagnosis commonly being performed on fine needle aspirates or core needle biopsies. Therefore, careful considerations need to be taken of the methodologies employed with the critical amounts of sample available. While FISH and IHC are slide-based with specific number of slides required to perform the assay already being established, generally 2 for FISH and 3 for IHC, or one of those slides is an H&E slide to determine the area of tumor, NGS requirements for successful tests become more limiting. They are based on the quality of the extracted nucleotide, the amount of extracted nucleotide, the size of the tissue, and the percent of tumor necessary is also a significant factor when determining the number of slides necessary for successful NGS results. Goswami and Associates estimated that the number of slides of paraffin-embedded tissue required for successful NGS could range from 1 when there is a large tumor with percentage on a large tissue to more than 10 up to 20 depending on the size and percent of tumor present. And when the number of actionable mutations is expanding, the amount of tissue collected is not. Pathologists are now increasingly required to exercise caution in specimen processing, conservation of tissue, and biomarker test selection to ensure that actionable information can be provided. It is therefore interesting to note whether or not, with the risk of unsuccessful NGS, it is worth using a large amount of tissue to determine those results, or pulling off a couple of slides for FISH or IHC. The other aspect to be considered is that the progressive nature of non-small cell lung cancer demands swift therapeutic response. Targeted therapeutic selection relies on knowledge of patients' genetic alterations, thus driving a critical need for timely results. CAP, IASLC, and AMP allotted 10 days between sample receipt and reporting of results. A 2017 assessment of real-world clinical practice showed that 34% of patients tested for ALK rearrangements had results delayed by more than 4 weeks, with the possibility that only 42% of those patients received appropriate therapy. In many cases, the time to obtain NGS results may take longer than 2 weeks. Extensive computer analysis and pathologist interpretation occurs only after the initial sequence analysis, thus delaying things even further. Even with technology continuing to advance and reduce the time to results, meeting those recommendations can be challenging for laboratories forwarding samples to centralized testing laboratories or performing in-house NGS. Risk of potentially delaying the placement of patients into proper treatment protocols continues. So is NGS the best option in identifying ALK and ROS1 gene rearrangements? Detecting breakpoints using DNA-NGS can be problematic. Breakpoints often occur in intron regions that can be difficult to sequence due to high GC content. DNA-NGS also requires prior knowledge of rearrangement partners. RNA-NGS heavily relies on the quality of RNA, limited when extracted from FFPE tumor samples. Some NGS assays rely on expression of imbalances between 3' and 5' portions of the gene to determine gene rearrangements, but they are assuming that the difference in expression is due to a rearrangement and not any other factors. So which methodology is the appropriate choice? Prior to the 2018 guidelines, EGFR and ALK were the only 2 molecular biomarkers recommended for non-small cell lung cancer testing. And most ALK testing algorithms used involve using either FISH or IHC methodology, and many including both at some stage. FISH testing has high sensitivity and specificity. It does not depend on gene fusion partners or kinase activity to demonstrate gene rearrangement. The probe design is critical in making sure that the more difficult rearrangements can still be identified. IHC testing has come close to matching the sensitivity and specificity of FISH. The potential need to confirm results with FISH for negative or challenging cases still remains. While NGS provides an array of genomic information, it is not always clinically actionable. It is, however, beneficial in treatment selection or when seeking clinical trials. Lacking in the level of sensitivity and specificity compared to FISH or IHC for the detection of gene rearrangements, NGS also takes a longer time to result and can delay patient treatment. Therefore, the question remains if the risks of invalidated outcomes and lower success rate are worth risking exhausting the sample prior to obtaining successful and actionable results. Again, the best course of action could be that there are advantages and disadvantages of each technology. Ideally, molecular laboratories would have the means to employ all the methods without any limitations. Most important to take into consideration is the patient's health and the limitations set on the available methodologies, the time they require, the sample availability, the effects of workflow based on the assay complexities, whether these affect the success rate of the assay, and the cost and reimbursement. Using a single test for all the answers is an appealing concept, but should the critical biomarkers that provide answers to actionable targets be prioritized and not risked? So what do we believe is the best course of action? With the current limitations of NGS testing, FISH and IHC have an advantage as preferred methods, specifically when it comes to identifying ALK and ROS1 gene rearrangements. However, the overall path of testing for non-small cell lung cancer patients can encompass all the previously mentioned technologies, where each has a role in the patient care life cycle, beginning with the initial diagnosis of non-small cell lung cancer, which relies on imaging like radiography and histological evaluation using H&E staining and various IHC assays, followed by prognostic as well as predictive evaluation to identify the optimal treatment using IHC, FISH, PCR, and even NGS, with NGS playing a bigger role later when monitoring the response to treatment or searching for options for the refractory or relapsed patient. As we discussed earlier, the non-small cell lung cancer genomic testing standards are expanding and evolving rapidly. Those involved in the patient care journey, the laboratorians, and the clinicians must be cautious when selecting new technologies, diagnostic approaches, and therapies. Laboratorians must ensure the quality and consistency of the methods employed when providing clinicians with actionable results. Identifying gene rearrangements such as ALK and ROS1 can still be challenging depending on the methodology employed. FISH is still an appropriate option, either as a standalone test or in combination with IHC, reflexing to FISH for borderline or difficult cases. While NGS may one day become the primary test method replacing others such as FISH and IHC, FISH can still enable laboratories to provide prompt and accurate and cost-effective results, thus facilitating the treatment decisions that allow patients to achieve the best possible outcomes in the disease that has been considered to have a very poor prognosis. And with that, I would like to thank you very much for attending, and please submit your questions and I will be happy to answer them.
Thank you, Yelina, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Yelina, the first question I have for you, do all NGS applications use libraries?
That's a really good question, and I thank you for asking that. In reality, the technology is always advancing, and the answer is no. However, those assays that are currently ongoing that do not create NGS libraries and go straight from the DNA or RNA that has been isolated are still in research laboratories and are still being developed and optimized. So when it comes to the clinical application of NGS, libraries are a significant benefit and often are even optimized specifically for the tumor, so providing targeted tumor profiles for patients.
Awesome. Thank you, Yelina. The next question I have, are there NGS applications that could identify gene rearrangements without prior knowledge of potential fusion partners?
There are, of course, those applications. With whole genome sequencing, you would be able to identify any fusion that has occurred. However, whole genome sequencing takes longer and it requires more incoming DNA samples. At the same time, as I mentioned in the presentation, RNA sequencing is an option, but RNA requires a higher quality of sample and is more sensitive to processing. And those are just two examples.
Wonderful. It looks like we have time for about one more question. Do you foresee NGS ever being the primary methodology used to assess molecular abnormalities?
I do, but just not right now. I foresee that being an aspect maybe in 5 years, but right now, even outside of non-small cell lung cancer, when evaluating any type of samples, whether it's hematology or tumor tissue, there is always a need to follow up, or there's always an aspect where the NGS results are just not quite there and do not provide all the information available. Therefore, it is still necessary to include other methodologies to identify gene abnormalities, especially when it comes to gene rearrangement.
Thank you again, Yelina, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Abbott Scientific Affairs Molecular Diagnostics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of your registration. LabRoots will alert you via email when this webinar is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay healthy. Goodbye.